Now let's take a look at a BLAST output. A BLAST output is presented in 4 sections. The first one is the program introduction. This gives the program name, in this case BLAST-P was used, the version number of the program, so 2.6.1 plus, and some particulars about the query sequence on this side. So, for example, the name of the query, that it's an amino acid, and the length of the query, 59 amino acids long. Some particulars about the database, or the subject the query was searched against. So in our case, we searched against the non-redundant protein database. And you also get the graphic summary, the descriptions, and the alignments. This is the graphic summary, the graphical summary that BLAST output on the web produces. It gives you an overview of the database sequences aligned to the query sequence. The query sequence here is represented in blue. And the database sequences are represented by the horizontal bars color-coded by score, shown by these colors up here, and showing the extent of the alignment on the query sequence. Separate aligned regions on the same database sequence are connected by thin gray line. It's not demonstrated on this particular example, but you may have seen it in other examples. Mousing over an alignment shows the database sequence title. Clicking an alignment displays a box with more details about the alignment and link to the sequence alignment itself in the alignment section of the report, which comes further down. If you scroll down the results page, you see the description section. This gives one-line summaries of the results. These are useful for identifying biologically interesting database matches and correlating this interest with the statistical significance estimates. The table gives a brief description of the database hit and its accession number, down here, e-value, percentage identity of the alignment, and the extent of the alignment, the database alignment to the database sequence. By default, the results are sorted by e-value in ascending order. You can sort the results by score, by coverage, the query coverage, e-value, identity, by clicking on the appropriate headers. Then comes the alignment section. This shows the pairwise alignment of the query with each high-scoring pair. It gives some details of the hit, such as a short description of the sequence, the organism it comes from, the accession number, which is hyperlinked to the GenBank entry, the total length of the sequence, of the hit, the extent of the alignment for both query and the database, the score and the e-value of the alignment, the percentage identity. The identity gives the percentage of identical amino acids or nucleotides between the query and the database, whereas the positives, which is the percentage of similar amino acids between the query and the subject. The line sandwich between the query and the subject sequence shows the consensus sequence of the alignment. Identical residues are shown as their respective letters. So if we look at this last HSP down here at the bottom, see the identical residues are shown as their respective letters, an empty space indicates a mismatch, and a plus sign indicates similar residues. In the case of a nucleotide-nucleotide search, you'd get an alignment output like this. The vertical lines between the query and the subject indicate an identical nucleotide match. A space indicates a mismatch. In this output, you'll notice that there isn't a positives count here, just a percentage identity count, and that there is a strand description of the alignment. The plus stroke minus means the positive strand of the query aligns to the reverse complement of the subject. And the letters in small case and grey color down here mark a low complexity region in this query sequence. So what is a low complexity region? It is a region of sequence composed of few kinds of elements, or low compositional complexity. They might give a high score that confuses the program to find the actual significant sequences in the database. They are marked with X for protein sequences or N for nucleotide sequences, and ignored in the BLAST program. Filtering can eliminate statistically significant but biologically uninteresting reports from the BLAST output. For example, hits against common acidic, basic, or proline-rich regions, leaving the more biologically interesting regions of the query sequence available for specific matching against database sequences. BLAST uses SEG filter for protein and DUST for nucleotide sequences. Okay, so let's move on to what other types of BLAST searches you can do. In addition to a nucleotide-nucleotide BLAST, which is done using the BLAST-N program, and the protein-protein BLAST, which is done using the BLAST-P program, you can do a nucleotide to protein search using BLAST-X. Here, you give a nucleotide sequence as a query, which is translated into all 6 frames and searched against a protein database. And you can do the opposite of that, which is give a protein sequence as a query and search it against a translated nucleotide database using T-BLAST-N. And within BLAST, you have the option of selecting between 3 different search protocols. For highly similar sequences, you'd use MegaBLAST. This is 10 times faster than BLAST-N. For more dissimilar sequences, you'd use the discontiguous MegaBLAST protocol. And you'd use BLAST-N for somewhat similar sequences. Within BLAST-P, you have even more options. Quick BLAST-P is an accelerated version of the BLAST-P that is very fast and works best if the target percent identity is 50% or more. SCI Blast, or position-specific iterative BLAST, is used to find distant relatives of a protein, and I will explain how it works in the next slide. The PHI BLAST, or the pattern hit initiated BLAST, uses a pattern or profile to seed an alignment which is then extended by the normal BLAST-P algorithm. The pattern is defined in prosite format. The pattern is used instead of the words that are usually generated for seeding alignments in BLAST-P. And Delta BLAST constructs a PSSM, or a position-specific scoring matrix, using the results of a conserved domain database search. and searches a sequence database. For all of these BLAST flavors, you can align 2 or more of your own query sequences by ticking this box where it says align 2 or more sequences. You will see, when you tick that box, you will see another box come up where you can input or upload your subject or your database sequence. So I mentioned SCI Blast in the previous slide. What is it and what does it do? So position-specific iterative blast, or SCI Blast, is a very useful tool for finding distant relatives of a protein. It allows the user to iteratively build a PSSM, or a position-specific scoring matrix. The first iteration is just a regular BLASTP search. This generates a multiple sequence alignment of the high scoring pairs above an E-value threshold and calculates a profile, or a PSSM. The PSSM captures the conservation pattern in the alignment and records it as a scoring matrix. In the next iteration, this profile is used instead of the original substitution matrix that was used in BLASTP, so as to detect sequences that match the conservation pattern specified by the PSSM. After every iteration, new sequences above the threshold are added to the PSSM. Now, this larger group is used to construct another profile, and the process is repeated till no more HSPs above the threshold can be found, or till convergence is reached. By including related proteins in the search, SCI Blast is much more sensitive in picking up distant evolutionary relationships than a standard protein-protein blast. In evolution, 3D structures of proteins may be conserved even after considerable erosion of their sequence similarity. SCI Blast has been demonstrated to be useful in detecting such relationships via sequence searches, which were previously only detected through direct comparison of 3D structures. In addition to flavors of BLAST I just pointed out, NCBI offers these specialized searches. Example, you can use Primer Blast to design primers to a template. CD Search allows you to look for conserved domains in your sequence. Using VecScreen, you can search for vector contamination. So please feel free to explore these in your own time. So finally, to sum up what we have covered in this session, I hope that from this session we have come to appreciate the complexity of the BLAST algorithm. What we covered is only the surface level of the full workings underneath the algorithm, but it should be sufficient to help you understand the sensitivity and specificity of your search. I hope that you now understand how the alignment score and E-value are calculated, and that the E-value is dependent on the size of the database and the length of the query sequence. The higher the bit score, the lower the E-value is, and that the significance of the E-value is determined by the query sequence and the reason for your search. And lastly, there are several different flavors of BLAST now available. Each have their own strengths, which you now know how to exploit for your research question. Finally, here are some links to resources on the content covered today that you might find useful. Thank you very much for your attention.